Hi, I'm Rajnish.
And I'm Bridget,
Welcome to Terra Science.
the podcast where reality matters.
Today we have the best students, biotechnology education and specialized training students. We spent the whole summer working on several different projects, and we recorded the first of the best episode on Terra Science 2 months ago. So here we are today. After 2 months, we are going to talk about all the fun stuff that the students did. Pratigya and Robert and Alexis, and we have Professor Krolikowski, where it all started. So, welcome, Dr. Krolikowski, to the Terra Science podcast.
Hey, great to be here.
So, let's start with tell us about how did the biotechnology program at Contra Costa College start, and then lead us up to the internship.
Right on. So, I'm a professor at Contra Costa College, and it turns out that Contra Costa College, based in Richmond, California, San Pablo as well, had the very first biotechnology training program in California. I guess it's over 30 years ago now. And so I was really lucky to be hired at Contra Costa College in 2007. After working at Mendel Biotechnology, which was a plant science company. And before that, I was on track to try to be a university professor, working in a postdoc, and before that in graduate school. And so I was hired to be part of the biotech program in 2007, and it's been the best job of my life. It's so great to have a place where we can have science accessible by community members. I came through real Ivy League kind of places, Rice University, Harvard University, and there's sort of this view among science folks that you have to go through all these hoops before you can do real science, and I do not believe that. And it's been so cool to be at Contra Costa College in a community college biotechnology program where people can start doing real science and make real contributions to um, useful products, useful discoveries, as they develop their expertise and knowledge through their whole education. And I think the CCC Best is proof that this is absolutely possible. I know this crew has been doing excellent work through the summer, and that everyone here is midway through their undergraduate education. And I think that's a perfect time to be making real contributions.
That's great. Well, so CCC Best is a collaboration between several institutions, actually, and the summer was quite exciting. Um, when when we thought about the Best program, what we were trying to do was to establish hands-on or direct science experience for the students that Dr. Krolikowski had in her biotechnology program. And we were fortunate. First of all, the Contra Costa College itself was excited about it, and they became part of the Best program, financially supporting it uh, to about one-third level. And then we combined that with the Carnegie Institution for Science at Stanford summer program, and they were also very supportive. So normally their summer program is a full week for 2 months, but they modified it for the students we had, and they made it 2 days for them for 2 months per week, which means that 2 days per week they got to go to Carnegie at Stanford. And then iCultiware, which is my company, we also had some funding for some projects. So we added the third contribution. So there were three institutions: Contra Costa College, iCultiware, and Carnegie Institution for Science. And so they all worked at three different places, and they worked on four projects, and it was a lot. We can see from their smiles how much fun they had. A lot of driving, for sure. But yes, so there were two industry projects. There were two products that we were testing, and actually one of them we are already writing a manuscript to submit, and I was going to announce and let you know today. And that this was Pratigya. Sorry, this was a little bit earlier before you joined, but Robert and Alexis, who worked on this particular manuscript, they're going to be co-authors on it, and we are sending it to Plant Physiology, which is a top journal.
Congrats.
Yeah.
That's exciting.
So this project was the seedlings. We were looking at how seedlings de-etiolate, and when we add a fertilizer, what happens. So that was one project. But then they had other exciting projects. They were they're doing immunoprecipitation to see what phototropin, a protein that is involved in phototropism, what that interacts with in vivo. And then we also had field trials for a bacteria that we are testing that can fix nitrogen. So I just wanted to kind of remind all of you again, all the work that you were doing, so that we can start to hear from you what your experience was, what you think you learned, and what you think how it will help you.
Yeah, and I just want to say, like, it sounds like the best program is just a really unique opportunity. And, you know, I'm from the outside looking in. I think it's really cool that you guys have put this together with, you know, so many opportunities mashed into the one, you know, internship. So I think that's really cool just to begin with. And thank you, Robert, Pratigya, and Alexis for joining us again. Um, I want to start off by asking you guys what your favorite memory has been this summer internship so far.
Yeah, I'll go first. So my most memorable is using liquid nitrogen for the first time to do RNA sequencing. Um, that that really brought the science into perspective. Like, but on top of that, you know, we were the ones who took all those samples, those DNA samples for the plants and the seedlings, and we saw, you know, on what level it takes to actually get those samples and how not to contaminate the samples as well, because they're very, very sensitive. And like, you know, with the light treatments that we did with those samples, like that was an extreme too. And you know, I only had prior experience of that in the Air Force working with night vision goggles, because you got to work in complete darkness with night
Oh
vision
wow.
goggles. And yeah, and so it brought that nostalgia back too, and like it just made me really, really excited, you know, to keep on progressing forward in my scientific, you know, endeavors and in my scientific career. And it's just a unique and a lifelong experience, and I was really happy to get to be able to do that with the team.
Well,
Awesome,
I'll
yeah.
elaborate just quickly on what he's talking about. So, in this experiment, we grow these seedlings on plates. They're only 3 or 4 days old, very, very tiny. And we have to expose them to dim light, like red or blue or far red, and in dark room. But the part that he's mentioning is you have to have liquid nitrogen, which is, you know, at minus what, 180 or something, and then harvest these in the dark, and then throw them in foil in the liquid nitrogen, so you can smell the liquid nitrogen. You are in this space. So you guys did really well. This takes a lot of practice, and there were many seedlings. We had 36 samples, 3 replicates of each, so 108 or something, right? it,
Yeah,
it was a long process, but you plated all the plates, you did all the work, you did very well.
thank you.
Okay, well, I would say for me, it's not a memory as of yet. The internship has been extended for me, which I'm excited about, and I'm also working on a couple new projects. I'm working with a PhyloStart project that's in conjunction with Dr. Krolikowski's lab, and so we're right at the beginning of that, and so I'm excited to see where she actually gave us an assignment where we're supposed to figure out how much of the project we can get done by Thanksgiving. She wants us to have like goals that we're going to
Good.
meet, and so I'm excited for that project to start. And then also we're resuming work on Dr. Khanna's plant consciousness project, which is a project that it's basically we're applying anesthesia to the stoma of
Right.
plants. It's a project that we started earlier, but then a series of unfortunate events kind of happened and we weren't able to continue on with the project. So that was one project down during the summer. But now that we're resuming, I'm excited to see the results from that as
Cool,
well.
cool.
But based on the
Just to
project,
clarify, it's not about plant consciousness. It's about studying consciousness using plants as
but,
substance.
yeah, but it's so much more exciting when you
Right.
describe it as plant consciousness.
I want to know
Okay.
what they're thinking.
But based on the projects that we did get accomplished, I think the immunoprecipitation project was my favorite. It was a lot of hard work, but I managed to work with one of the scientists down in Dr. Wang's lab very closely. And from that interaction, I learned a lot of things. In the protocol itself, it was, I wouldn't say it was a big protocol, but it was kind of complex. So, some new techniques that I haven't really seen yet. And so, based on the ability to just learn new things, I think that the immunoprecipitation project was one of my favorites.
Cool. Thanks, Robert.
Okay. So one of the favorite memory that I have from this internship was the iron extraction too. It was like we get to work with the team, and we were like all like helping each other, and six of them, like Dr. Khanna and John and Dr. McHugh and all three of us, we were like working in a team, and that was like at the end of that, like because of our good teamwork, we got good results. And another memory would be from Carnegie, like when we started to build our own lab, like create our own space. We did a
Oh,
lot of
cool.
work there to create our own space because it was a like old building,
Mm-hmm.
and a lot of like research has been done in that like lab, and then we got to work there. We got to create our own space and work there, so that was the best thing.
Fantastic. That's really
Yeah,
cool.
the RNA extractions that we all did, which were, like I mentioned, 36 samples. It was six of us, took us 2 days full time, and they are now being sequenced. We will have the sequences probably next week, but they all passed QC. They all look good. So far so good.
Even though I wasn't part of the team, I'm starting to build memories of working with the best
Aw.
in our, at Contra Costa College, we have this brand new, amazing science building. And as part of that, one of the really exciting parts of that building is a brand new biotechnology lab. And it's just packed to the brim with equipment. Some of it we've always had in the program, and some of it was newly purchased as we have moved into this new building. And I want to give a big shout out to Brian Williams, who's the technician that cares for that space and makes everything possible, the classes I teach, and most importantly, this collaboration with Rajnish. It's not every person who would be willing to do a little extra work to make it so a whole team of students could use this amazing space in a way that's not a regular class. And it's just, I'm really, really excited to have our community college space not just be for classes with grades, but all this fabulous equipment that we use for um, teaching can also be
Mm-hmm.
used doing real projects for iCultivar. And it's really, really an exciting thing to see in that space. And I think it's going to spur that kind of collaboration even beyond science at our college. So it's really just wonderful to work with Rajnish on creating something bigger and better at the place that I work.
So wonderful. Yeah.
Also, I want to add that I think Brian makes things work really smoothly. We don't really have to worry about all the things that are needed in the lab because he's taking care of all of that. So, yes, big thanks to Brian for that.
Yeah. And Raj, what's one of your favorite memories? So working with the three of them too.
My favorite memory working with them, I think, is has to be the poster session because um, we knew that at the end of the internship there was going to be a poster session that Carnegie always does at the end of the summer internship, but it kind of slipped our mind, and we were just sitting and going through our data and stuff, and oh, it's like in 4 days. um, and we were not even at Carnegie because that's where the big poster printers are. So I think the 2 days before the poster session, we were there. We sat in the nice on the picnic tables in the back of Carnegie, made the whole poster and in the morning, and it was sent. We had already passed the deadline of printing, but they were nice and they helped us print it.
Oh.
But then the best part was there were so many posters. There were all these other interns, and all the other interns did their 100% of their internship at Carnegie.
Mm.
But our interns, best interns, were all over. So I let them tell us what was how it was received. I got very positive feedback from everyone, from how they explained what the research was. But tell us what some of the people were telling you.
So they said, how can you guys work all over these places? Did you guys just do it in the summer? Like they were like shocked that we did all the project within the summer.
Yeah, because there were
It's
four
impressive,
projects at four
yeah.
different locations.
I would kind of just piggyback off that point. One of the major, I guess, critiques that we got was just how did we manage to do so many projects? And for me, I didn't know any better. I think that the poster session kind of encapsulates my experience in biotech just in general, where, like I mentioned on the last video, my start with biotech was with through Dr. Krolikowski's Urban Tilth project, where um, I can't say this for certain, but I think that it was a lot of upper level techniques that we were learning in that class, and so I kind of just jumped in head first. And so with the poster session, because I didn't know any better, because I didn't realize that many other people would probably take on one project at a time. No, we were given four, and actually five, because like I said, we started the quote unquote plant consciousness project, but we didn't manage to finish it. But it's not for a lack of trying. I came down with COVID. I think the person who was doing the microscopy, he came down with COVID. So there were a few different things that happened, but it, like I didn't realize that taking on such a load was something that would be unique. But like you get what you get.
That's also Rajnish's expertise, is doing a lot and handling it somehow. So I'm
Well,
glad.
my rule of thumb is that you do 10 things and at least one of them will work. So,
you, there you go.
yes.
How about
So,
Alexis? Any thoughts from your poster experience too, or no?
Well, it's just the questions that our team got were a lot more difficult than compared to the other poster presentators because we
Mm.
had so many more projects, compared to them. And so, there's people there that are postdocs and, there's people there that already have their PhDs and stuff like that. And so the level that the questions were above our head and so that's where Dr. Khanna would interject and, their conversation, we would be like, oh my God, they're just, taking this to the next scientific level. And, that's what it takes though is, you're sharing this poster and this information with everybody else there. So, hopefully they can incorporate it into their research as well, or it can open up their mind to like, oh, we can actually help them with this part or this aspect of that research. And so it was
Mhm.
a really enlightening experience.
And it sounds just really cool that you get to do so much in such a short amount of time, because like what Katie was saying earlier, to get to those higher levels in the science field, it sounds like you have to really take it slow in that older method of going through internships and finding a job per se, or going through the school. But just to be thrown in there and get to try it all, or just experience different types of research, it just seems like really a good opportunity to see what you do like, what you don't like, what you're good at, where you really need to learn. So maybe that's also something that's like a benefit of having all of those things packed into one.
That's a good point,
Yeah.
Bridget, and I want to add something there. I think it's not just that these students, were there and they had this opportunity. I think a lot went into designing
Mm-hmm.
the biotechnology program itself, the courses that Dr. Krolikowski has designed. I think they offer opportunities to students who are interested in developing it further. They are more. They're not, just a teaching method where this is how it is, this is how it's done, but I'm become familiar with her course style, and they are really open. They're more free for students to explore in different ways in how they want to explore. Just like now, Robert was telling us, she asked them to tell us how much will you accomplish by Thanksgiving. She didn't tell them this is what you have to do,
Mm-hmm.
because they are learning how science happens, so that they are in charge during the course. It's a really, really unique course. I've never ever seen anything like this. But this is why it prepares them to take up these challenges, because they've already learned how to manage
Mm-hmm.
science and their time. So I just want to say thank you for putting so much effort in developing these courses.
I appreciate that feedback, Rajnish. I like I said, this is my favorite job I've ever had, and I love teaching. And the mission of our college really is to help students reach their own goals. And part also a mission of community colleges, I think, is to make sure that people from all different experiences, all different walks of life, all different cultures, all different levels of opportunity before they arrive at our space. Our job is to help make sure that the world embraces the wisdom, knowledge, and brilliance of the full diversity of our population. And I think a lot of times the folks in charge of how things go are only from a certain segment, and that's true in all different experiences. But to me, science needs all kinds of ideas being added to it. I'm just driven by that. I've been reflecting on the people I admire most in science, and of course Rajnish is one, because he's just doing something that no one else does. He's running a company and collaborating with 15 universities and a bunch of other companies. But before that, the folks I admired were often women scientists. Many people might know of Barbara McClintock, who discovered transposons. I when I was a After I was a community college teacher, I saw a talk by Elizabeth Blackburn, who learned about telomeres, the ends of chromosomes. And one of my mentors was Nancy Kleckner at Harvard University. And all three of them met with incredible resistance because they thought differently. And they're all just brilliant people who ended up showing people a whole new way of thinking about genetics. Those three folks are all about genetics. And I think it has touched me throughout my whole life that it's so important to keep pushing when you know you have a new and creative, different idea. But it's also struck me how hard it is to bring new ideas into any system. And science is so important for us as humans. Like, we rely on science to find out how things work. We rely on science to bring new technologies to solve incredible problems on this earth. And if we don't make it so that new ideas can come in, we're not going to solve the problems. And so to me, students like the three folks you see here are the people who are going to save the world. And this sort of accelerated way of jumping into the scientific establishment is one way to make sure science benefits from all these different perspectives. And so it really means a lot to me that this program exists, because it shows that it's totally possible to change the world in the middle of your undergraduate education from community college. And I think it's totally true. I see students every day that can totally be in a spot and work with Dr. Khanna. And I'm so glad that you three are paving the way, because this is a great future.
That's amazing. And I think with that note, it's a perfect segue to ask what's next for each of you guys. Like, what, are your plans now that your internship is up, or Robert, you're still doing it, right? But how do you think the experiences you've had this last, couple months will help in the future? And, what are your plans to move forward?
So I've already encountered like where it's taking me because I just transferred to Cal Poly Pomona and I'm meeting people who are trying to be biologists. I'm meeting people who are going to be botanists and all that. And I share with them the experience that I had with the iCultivar internship, and they're just so amazed that there was an opportunity like that even available. But the institutions we got to work with, the USDA, Stanford, like UC Berkeley, like they didn't know that there was an actual program that collaborated all these places together. But the fact that it was at the community college level is what they were even more surprised about. And where I'm taking it to is like I'm going to incorporate this into my research that I'm going to do in the future, because I have to do a senior project for my graduation to get my degree in chemical engineering. And so I want to incorporate the research that we've done in iCultivar, into my senior project. And, just the experience that I've gotten from iCultivar is what's, helping push that forward. Because if I would have never joined this internship, I've never would have gotten that lab experience that from, because we're doing research on an international level on some of our projects, because we're not just helping the community, we're not just helping, our own country, we're doing research on drought, and that can, have really amazing effects for, countries that are suffering with their plant and, keeping up their crops and all that, especially what's happening now with inflation. And so, it's just it's given me the drive to move forward, but it's also shown me, what it means to be a researcher. It's shown me what it means to be a scientist. It's shown me what it takes to incorporate your research, your knowledge to actually help everybody around you. But, it takes a team to do research. You can't do anything by yourself. And that was the most important part of this whole experience is you have to network. You have to go out there. You have to put yourself out there. You have to put your research out there. And if you're afraid of being peer reviewed, then you probably there's probably something wrong within your research, and you have to go back and you have to relook at your notes, and you have to relook at everything that all the data you've taken down to make sure that you are 100% correct, because someone is going to go behind you and try to do the same research that you did or peer review the research that you've done to see if there's any faults in there. And that's the beauty about science is we're all checking each other to make sure it's the most absolute and best information available to everybody out there in the scientific community and possibly the world if they want to look into it.
Well, that's great, and all the best to you with your new venture there. And of course, we'll stay in touch because there'll be results and things that we'll be communicating. So you don't get to run away by any means. So we'll definitely stay in touch, and all the best of luck to you.
I wasn't planning on running
Pratigya,
anyway.
Pratigya, you want to go next?
Okay, so the plan, like what I got out from this internship was how to work in a good team, how to work in the labs, how to like handle everything. And I'm not sure what I want to choose my major for right now, but it was really good experience to like know all the lab stuffs, how to do it, how to work with collaboration, and how to handle everything, every sample with care. And
I will add something there. I think Pratigya was one of the very early students who joined us. She had just started in the program, and I think it was a lot of information. Many of it that you have probably not even encountered before this.
yeah.
So so it was kind of an explosion of science in the 2 months. And but you did well, and hopefully that will set you or prepare you for the courses that you're taking, biotechnology courses you're taking at Contra Costa, and continuing with your studies. And we definitely intend to repeat this every summer, this intense internship. But I also wanted to mention this internship runs throughout the year. So it's not just the summer internship. we reduce the amount of time we spend because students have other responsibilities during the fall and spring semesters. But Robert, like you mentioned, so before we went to him, I wanted to just give a little bit of background where he is currently. So he's continuing with the internship, but in an exciting way. So there is a project that we are starting, which is PhyloStart, which are bacteria that we are developing for commercial application on greenhouse grown tomatoes. And this work is being through collaboration with Dr. Krolikowski and Dr. Britt Koskela at UC Berkeley. So Robert is going to be continuing on that along with some of the other projects, which of course never end. So he's still continuing with all four plus five and the six. But the exciting opportunity here for Robert, I think, is from community college. We met with Dr. Koskela, and I think and I hope Robert is interested in eventually starting to transition to UC Berkeley to complete his graduation. Okay, Robert, now you can tell us.
Well, now that he gave me the lead in, I guess there's a short term goals and then the long term goals. I think Dr. Khanna kind of expressed some of the long term goals that he has for me. The short term would be, of course, to finish out the semester, get knee deep in the PhyloStart project that we're doing. I can't wait for that to really get going. Like, so when we get into the science of that. Oddly enough, now that I'm in that class and I'm working with, the class have been split into smaller groups, so subgroups, and the group of people that I'm working with, I think that uh, there are four. Two of the four, they're kind of just been recently introduced to biotechnology as well. So they're kind of in the space that I was when I started. And they have a lot of the same questions. And I'm like, oh, I know that face. But now I get to act almost in a, I don't want to say a mentor, but like I can kind of direct the questions that they have towards some of the goals and that Dr. Krolikowski had for me when I started with Urban Till. So that's very interesting how that came full circle for me. Um, Also, with some of the tomatoes that we had harvest, we were growing over the summer. We're starting to harvest those, and I'm working with Dr. Bogamoni down at Carnegie with lycopene measurements. So, like, just completing the smaller projects that we've done. I can't wait to bring all of those projects to fruition and see some of the data. But on to the long term goals. I'm very excited about the connection that I've made to UC Berkeley and Dr. Koskela. Maintaining that connection is very important to me. And through the PhyloStart, she has my name. I've met her recently, so she has my face, and now she's going to have my work. So I don't know. There are a lot of good opportunities opening up for me. I'll just say that.
Fantastic.
Great.
Well, I guess we can give a special thank you and shout outs to some of the other people who have been working alongside this project. So, Yang down at Carnegie Labs, Julia and Asa, who worked
Mhm.
alongside us at the USDA. Um, I think his name Elijah, right? I've
Mhm.
only met him once,
Yeah.
and so he's doing work on the from UC Berkeley as well. And I feel like there's one more person that I'm forgetting to thank.
Sambidan.
Well, yes, Sambadan as well. I wish you guys could have met him. But I feel like there's one. Oh yeah, the guy who's in charge of the greenhouses also down at Carnegie. I can't remember his name for
Johnny.
some reason.
Yes. No, no, thanks for bringing all of these things, all of these people. So yes, Julia and Asa, who work with Dr. Koskela at UC Berkeley, and then the people, the staff, greenhouse staff at Plant Gene Expression Center, where we rent the space, and also the greenhouse staff at Carnegie, who they all work directly with you, and you work with them to keep things moving smoothly. And thanks for bringing that up, Robert, because I think it's also important for us to thank the other professors and other people that you interacted with. So Dr. Xiong Wang, who was instrumental in helping establish this new program, the summer program, and I think he was excited hearing about your poster as well. He spent quite a bit of time there with you, Robert, right, on the
Yeah.
poster. So Dr. Xiong Wang, Dr. Roberto Bogomolny,
Yeah.
who's teaching you biochemistry, and what an amazing person to learn that from. Dr. Roberto Bogomolny was a PhD student of Melvin Kelvin, and you're learning directly from him. So it's really great. And of course, other people that we've been collaborating with, John Farrell, and he's the vice president of Azomite. And Azomite has been very supportive of the internship and has also contributed financially. So we definitely want to thank Azomite and John, and many other people. I think during your work at Carnegie, you got help from, like you said, Yang. There's so many people who you interacted with.
Yeah.
So yes, thank you to all of them for this.
Just want to say thank you guys so much for joining us on the Terra Science podcast. If anybody's listening, please make sure to like and subscribe, hit the notification bell. If you're listening to us on Apple, leave a review because that really helps. But overall, we appreciate you guys being a part of this episode. And yeah, we'll make sure that everything about the BEST program is down in our description below. See you in the next one.
Thank you, Bridget.
Bye. Thank you.
Everyone.
Nice to see you again, Alexis.
Yeah, it's nice seeing everybody.
Well, let us know.
You've been watching Terra Science, the podcast where reality matters.
We discuss food, planet, consciousness, the issues that we face, and the solutions that can be offered. And we discuss with wonderful guests who are leading the way in finding these solutions.
Don't forget to like and subscribe and hit that notification bell.